It's a pleasure to be here. I will talk about next slide. Autophagy and obesity, and as a new molecular link that may obesity accelerate aging, and autophagy that may prevent the aging process to some extent as a cytoplasmic rejuvenation mechanism. Next slide. So I quickly pass my disclosure information, which is not relevant to this talk. Next. I just would like to introduce our interest in the question how pharmacological, genetic, or nutritional manipulations can actually increase health span and lifespan in model organisms. And next, as a partial answer to this question, we found that next, please, autophagy has to be induced next to mediate this process. So frequently this occurs by pharmacological or nutritional ingredients that next stimulate autophagy and that we call caloric restriction mimetics. So they can, through the induction of autophagy, which can be proven genetically, increase health span and lifespan in flies, in C. elegans, and to some extent in mice. And what is interesting is that when we test these agents that prolong lifespan in model organisms in mice, we see that they systematically have anti-obesity, anti-diabetic, hepatoprotective, cardiopreventive, and oncopreventive effects. So they have a very large impact on different organ systems. Next slide. So recently we came into a story that is dealing with ACBP, acyl-CoA binding protein, that according to the literature is a protein, next slide, that is released by unicellular fungi. So it's the only protein that, for instance, is released by yeast, Dictyostelium discoideum, during autophagy. So it's an autophagy-related release. And ACBP actually has been knocked out in C. elegans and in yeast and increases longevity. So it is a protein that in a way is bad for lifespan. And I will dwell on this question in different mouse models later on. So next, ACBP is phylogenetically conserved. It's a small 90 amino acid long protein that has two names. Next, ACBP as an intracellular protein that is participating in lipid metabolism, as the name indicates. And as an extracellular protein in mammalian system, it is called DBI for diazepam binding inhibitor. It acts actually on the only inhibitory neurotransmitter receptor, GABA-A, which is not only expressed in the central nervous system, but close to all of our cells outside of the central nervous system. And next, the intracellular protein can be released through autophagy, not only in yeast, but next also in mouse and human cells. So in vitro, we can culture human cells or mouse cells, keep them in nutrient-free conditions, NF, and then show that the intracellular signal corresponding to ACBP by immunofluorescence vanishes as the protein appears in the supernatant of the cells. And very similarly, next, if we use the most physiological mechanism to induce autophagy in mice, which is a starvation period, for instance, for 24 hours, all the cells in the body will practically mount an autophagic response that is linked to the disappearance of the fluorescence signal, here for instance the liver, but this is seen for any other organ, and the appearance of ELISA detectable ACBP DBI in the plasma and in extracellular fluids. And next slide, when we inhibit autophagy, we can prevent this process. So we can safely say that even in the mammalian system, autophagy, which is a mechanism of cellular renewal, is linked physiologically to the secretion of ACBP DBI, which occurs in a unconventional Golgi-independent pathway. Next. So we asked whether ACBP-DBI might itself have an impact on autophagy. It turns out that it actually limits autophagy in vitro experiments. So if we measure, for instance, the lipidation of LC32 by Western blots, which is a marker of autophagy, we can show that the neutralization of extracellular ACBP by suitable monoclonal antibody induces autophagy. Or next, if we add the recombinant ACBP-DBI protein to the cells, especially in unfed conditions, we see an inhibition of autophagy. And we can show this also in vivo. So if we measure autophagy in this example, next slide, in the liver, we can show that injection interperitoneally or intravenously, so peripheral injection of an antibody directed against ACBP will induce autophagy, while injection of the recombinant protein will inhibit basic autophagic flux or autophagy induced by starvation in the mouse. And this can be found in the heart, in the liver, in the muscle, wherever we look at. Next slide. So we can say that not only autophagy releases ACBP-DBI, but in addition, next, ACBP in the extracellular medium will mediate a paracrine feedback loop to limit autophagy. And next slide. So we set out to do some additional next physiological exploration. So for instance, if we inject recombinant ACBP-DBI interperitoneally or intravenously, we get an immediate increase in food intake in the mice that is correlating with an increase in body weight, at least in the short run. We see signs of lipoanabolism. So if we do whole body respiratory studies, we see that ACBP causes a decrease on fatty acid consumption and an increase in adiposity in tissues, as well as a slight reduction in glycemia. And this is important for the mode of action of ACBP-DBI with respect to food intake, as we will see later. Next. On the contrary, if we inject a neutralizing antibody peripherally, we see an immediate decrease in food intake, especially unfed conditions. So usually become very much interested in food intake when they have been starved for 24 or 48 hours, and they lose interest in food if they receive this neutralizing antibody. At the same time, they show the starvation-induced reduction in glycemia is prevented by neutralization of ACBP. And we also see an increase in fatty acid oxidation. So lipocatabolism. And next slide. So we believe that these data can be interpreted in the sense, next, that ACBP-DBI, once it is released, we call it now the hunger protein, so HP, causes immediate food intake, eating behavior, and this closes a sort of neuroendocrine next feedback loop that removes the primary cause of autophagy in this model, which is starvation. And so, next slide. We asked the question whether ACBP-DBI would be perturbed in humans with disorders in the eating behavior. Next. So we actually observe that in anorexia nervosa, circulating ACBP-DBI levels are very low. In obesity, they are high. Upon bariatric surgery, they tend to go down again. And if we, next slide, perform an aggregate analysis of the correlation of body mass index with circulating ACBP levels, we find a very nice, highly significant correlation. And next, so we looked at the correlation of ACBP with fasting insulin levels and transaminase concentrations, and we do see positive correlation, as if ACBP DBI would be a biomarker of prediabetes and subclinical liver damage linked to NASH. And next, we looked at the mRNA levels that are upregulated in obese individuals and that can be partially corrected by putting overweight and obese people on hypocaloric diet or by bariatric intervention. So there is an impact of the whole body fat consumption and calorie consumption with the increase in ACBP DBI levels. So we can see next slide is also in mice. So if we deal with OB mice, which are spontaneously over-consuming a normal diet, or if we feed mice a high-fat diet, we systematically see an increase in mRNA levels of ACBP in tissues, an increase in the protein concentration in tissues and in the circulation. And next slide, please. So we think that this whole body weight rheostat that involves ACBP DBI, the hunger protein, is probably also conserved in humans. So we did some additional studies next to show that next, please, that yeah, in anorexia nervosa, I already showed this. Next, please. Next, that we can overexpress ACBP DBI as a transgene after hydrodynamic injection of constructs that will express this factor in the liver only. You see that this is sufficient to increase the concentrations of ACBP DBI in the plasma, to increase cumulative food intake, and to increase weight. And next slide. We can perform a whole body inducible knockout that is tamoxifen inducible, and this will again, as the example for the antibody, maintain glycemic levels in unfed conditions and induce an unfed-like metabolome with an increase in fatty acids and ketone bodies in the circulation. And next slide, please. If we perform this inducible knockout, we can show that in all imaginable conditions, weight loss is favored. So in unfed conditions, ACBP knockout mice lose more weight. In a high-fat diet, they put on less weight. And if we switch this high-fat diet to a normal diet, they lose more weight than control mice. And next slide, please. The neutralization of the hunger protein ACBP DBI does affect central appetite control. So even if we inject the antibody into the peritoneal cavity or intravenously, 30 minutes later, we do see the inactivation of orexigenic neurons in the lateral hypothalamus. And next slide, please. At the same time, the activation of anorexigenic neurons in the ventromedial nucleus. And this is significant both in unfed and fed conditions. Next slide. So I don't have really time to go through all the mechanisms. It appears that an elevation of ACBP will induce the rapid expression of glucose transporters in liver and other cells, sucking into the cells the plasma glucose, which declines. And this is then indirectly responsible for the activation of orexigenic neurons and a hyperphagic response, overeating. If we neutralize ACB, we reduce GLUT expression and at the same time increase lipolysis in the white adipose tissue. This causes an increase in plasma glucose levels that will cause the activation of anorexigenic neurons and appetite control. And next slide, please. Next, We can even perform an autoimmunization to break self-tolerance against the hunger protein. And this again, it causes a long-term effect that improves metabolism, favors body weight loss, reduces body fat. And next slide, please, has a positive impact on whole body composition that you here see by computer tomography. And next slide will reduce adiposity with a reduction in the mean diameter of adipocytes. And next slide have even an impact on the diabetes-related glucose tolerance test that is positive. Next slide, please. Reduce signs of hepatosteatosis that can be measured histologically. And so to summarize, it appears that very different ways, next, different strategies to inhibit the hunger protein next, namely the knockout, the passive and active immunization, the injection of monoclonal antibodies or the breakage of tolerance will have similar effects that are globally next, a reduction in lipo anabolic reactions and increase in lipocatabolic reactions with a tendency to prevent weight loss in the context of obesity at many levels. So we believe, next slide please, that we have discovered a novel body weight rheostat that I already mentioned. Next, it is important to note that ACBP DBI has very similar functions through evolution. So for instance, in yeast, it stimulates sporulation, which is the only way for yeast to look for new food resources. So it's an appetite stimulator throughout phylogeny, from mouse to flies to worms and to yeast. And it is very interesting, next slide please, from the point of view, next, that it is one of the few orexigenic stimulators that is actually activated and elevated in human obesity. Most of these stimulators are actually reduced paradoxically or not changed. Remember that ghrelin, which is a well-known appetite stimulator, is paradoxically reduced in obesity. It makes no sense, but ACBP DB1 does, because it is increased in obesity. And finally, I would like to, next slide please, mention the reasons why we should utilize ACBP DBI, not only because it is phylogenetically linked to aging in model organism, it is also because it is a logical target, it is a peripheral regulator of appetite that is not causing depression in mouse models. It induces autophagy, it has a broad pro-health effect, including at the comorbidities of obesity, diabetes, and NASH. And so next and last slide, I would think that we can use antibodies against ACBP DBI as autophagy checkpoint inhibitors for future interventions on obesity and obesity-related diseases. Thank you for your attention.
Guido, thank you so much. Professional, right on the dot at 11:30. Thank you so much.